Hello everyone, the article that I'm going to talk about today is a research done on the transcription factor TPRFX1, which is an essential regulator of amylase and cellulase gene expression in Saccharomyces pinoculus. Saccharomyces pinoculus is a fungi present in plants and produces integrative amyloidic and cellulitic enzymes. These enzymes are required for growth of Saccharomyces pinoculus. The picture in black and white shows the structure of Saccharomyces pinoculus under a microscope. Yet we need to analyze the transcription factors or TFs that regulate gene expression of these enzymes and their system because TFs plays a critical role in controlling gene expression by binding to specific regions of DNA and promoting or inhibiting transcription. Thus we can understand the regulation of gene expression and we can also identify the regulatory network since TFs don't act alone. This analysis helps us to identify other genes or proteins that are part of the regulatory system, and it also provides valuable insights into cellular response, drug target identification, and biological pathways. Now let's give a brief overview on the enzymes. You can see the structure of amylase. Amylases include different branches that attack the glycosidic bonds of amylose and amylopectin, which is a starch, and degrade the starches. After degradation, it generates varying lengths of straight chains and branch oligosaccharides. Here we can see the structure of cellulase. Cellulases are a type of fiber that also include different branches that attack internal glycosidic bonds of cellulose chains to release chain ends and result in soluble products into glucose. Lastly, you can see the structure of regulatory factor X or RFX family proteins here. It regulates both cellular differentiation and cell cycle and contains an RFX DNA binding domain. In this study, they screened and identified novel TFs that regulate the expression of amylase and cellulase genes in T. pinoculus. They described a key regulatory gene, Saccharomyces pinoculus regulatory factor X1 or TPRFX1 or delta TP0612A, which encodes an RFX protein that directly regulates the expression of amylase and cellulase genes. They also used fungal strains in this study. These strains include the parental strain, delta TPQ70, and complementary strains CTPRFX1. Through this research, they followed multiple steps and found valuable results after each. First, they maintained the fungal strains on potato dextrose agar or PDA plates at 4 degrees Celsius. Incubation step was followed afterwards. The incubation temperature for the fungal cultures was 28 degrees Celsius. The researchers extracted both DNA and RNA from the fungal mycelia harvested from PDA plates where liquid media was lysed using a specific lysate reagent. Total DNA was separated from the lysed suspension by removing proteins, and the total RNA was extracted using an RNA kit. To delete the specific genes in T. pinoculus, deletion cassettes were created using a technique called double-joint PCR, which is a PCR-based molecular tool for gene manipulations in filamentous fungi. The cassettes were amplified using specific primers and then introduced into T. pinoculus cells that lack the TPQU70 gene. To complement the mutant strain lacking the TPRFX1 gene, a gene complementary cassette was constructed using a technique called fusion PCR, which is a simple and easy method to produce fusion DNA fragments without the need for restriction, enzyme digestion, and DNA ligation. The cassette was created by amplifying these DNA sequences using specific primers. The resulting cassette was then introduced into the mutant TPRFX1 cells. For cellular hybridization, first the genomic DNA of the delta TPRFX1 strain was extracted and digested with a specific enzyme called Sac-I. The resulting DNA fragments were separated on a gel and then transferred to a nylon membrane. A special kit was used to detect specific DNA bands on the membrane. This was done by using a labeled DNA probe that was created by amplifying specific DNA sequences using specific primers. As a result of this research procedure, they found that the 7 novel TFs are present that regulate amylase production in T. pinoculus. Out of the 7 TFs, delta TP0612A showed a significant expression level compared to parental strain delta TPQU70. This was confirmed by cellular hybridization analysis and means that TP0612A is the most important regulatory gene. Then the researchers aimed to assess the activity of enzymes, specifically amylase and cellulase, and determine the concentration of proteins with fungal mycelia. After performing RT-qPCR analysis to measure gene expression levels and purify the specific DNA binding domain, they used a SMART analysis and they found that TP0612A encodes an RFX protein containing an RFX DNA binding domain. To determine enzyme activity, the researchers used various substrates and incubated them with diluted enzyme solutions. For amylase and cellulase activity, different substrates such as soluble cornstarch or SCS or wheat bran plus Apcel or WA, which is a carbon source, were used. After incubating the reaction mixtures at specific temperatures and times, they found that TPRFX1 is required for amylase and cellulase production in T. pinoculus. For RT-qPCR analysis, the researchers used the PrimeScript RT reagent kit. They performed PCR reactions including forward and reverse primers, cDNA, and sterile water. The PCR reaction involved several cycles of denaturation and annealing or extension. The fluorescence signal was measured and the relative expression levels of the tested genes were calculated. Lastly, the researchers expressed and purified a specific DNA binding domain called TPRFX1190376. They amplified the DNA sequence encoding this domain using PCR and cloned it into a vector. After purifying the recombinant proteins, they found that TPRFX1 regulates the expression of amylase and cellulase gene in T. pinophilus. Results also indicate that the DNA binding domain sequence is targeted by TPRFX1 and TPRFX1 regulates amylase and cellulase gene expression by directly binding to their promoter region. In summary, the researchers aim to identify and understand the role of TFs in regulating the expression of amylase and cellulase genes in the fungus Saccharomyces pinophilus in this study. They used comparative transcriptome and genetic analysis to screen for novel TFs involved in this process. In this study, the researchers identified a TF called TPRFX1 belonging to the RFX TF family as a positive regulator of amylase gene expression. RFX1 proteins are found in various organisms and are involved in immune responses, cell differentiation, and other processes. The researchers found that TPRFX1 directly binds to the promoter region of amylase and cellulase gene, which contain a specific DNA binding sites for this TF. They discovered a DNA binding sequence for TPRFX1 that shares some similarities with the consensus RFX binding sequence found in previous studies, which are the nucleotides T and the 5 nucleotides G and AAC. They also observed that TPRFX1 has additional regulatory roles in T. pinophilus. It positively regulates growth, and its deletion leads to reduced transcription of important amylase and cellulase genes. This decrease in gene expression results in impaired cell differentiation. As illustrated by the graphs, we can see that delta TPRFX1 accumulated similar amounts of mycelial biomass as that by delta TPQU70 in glucose medium, whereas the mycelial biomass of delta TPRFX1 was slightly lower than that of delta TPQU70 in SCS medium, suggesting that lack of TPRFX1 affected the utilization of starch by T. pinophilus. Therefore, TPRFX1 controls T. pinophilus sporulation and growth. This research focused on identifying novel TFs that regulate amylase production in T. pinophilus. However, it is important to note that only 7 TFs were analyzed in this study. Future work could involve a more comprehensive analysis of other potential TFs that may also play a role in amylase regulation. Also, this study found that delta TP06128 shows significant expression levels compared to the parental strain. In future works, it would be beneficial to further investigate the functional roles of other 6 TFs identified, because understanding the function of these TFs could provide a more comprehensive understanding of the regulatory network involved in amylase production. Amylases and cellulases are important enzymes used in various industrial processes such as biofuel production, textile processing, and food and beverage industries. Understanding the regulation of gene expression can help optimize enzyme production, leading to more efficient and cost-effective industrial processes. Cellulases are crucial for the breakdown of cellulose, which is a major component of plant biomass, into simple sugars. These sugars can then be fermented to produce biofuels. Amylases also play a role in starch metabolism in plants, as cellulases are involved in cell wall remodeling. This knowledge can be utilized in crop improvement programs to increase biomass production and improve plant resistance to pests and diseases.